tomorrow in lab, you'll be doing several things. These are kind of foundational skills in a microbiology lab. So if Duncan already emailed you, great, but expect an email from him. If he hasn't, he's going to be asking you to look things over, the PowerPoints that are posted. So you go to my EduCat page. Duncan and I are sharing that page, so his lab stuff is in that page too. So it's a one-stop shopping. Just go to the EduCat and you'll see the lab section by week. I think he's going to arrange it. So look at that, please. So you're going to be learning the streak plate procedure, the spread plate procedure, and a crosshatch procedure. And so it's 2 hours, and we're going to need to be like on task to get all this done. So if you read some of those PowerPoints ahead of time, you'll at least have a, somewhat of an idea of what you're going to be doing. And it's something that you'll be using throughout the semester, and probably when you get to your independent research, you'll be using it as well. So make sure that you're really trying to get comfortable with it. It's going to feel awkward if you've never done it. Some of you, I know who've had Gen Micro or Med Micro, you've done this, but it's a good refresher. But if you haven't done it, it's going to feel awkward. It's just a new physical skill that you have to do, and it's using delicate tools for some of that. So just embrace it, go with it, and slow down, and don't rush through it. Okay, don't forget about your field guide ideas. The weekend coming up, you could go hiking, go looking. Got any questions on the field guide idea? Yes. How far? I mean, I've gone up onto the Big Bay Road a fair distance in the past. Sometimes out toward Nagani area. So it depends. I mean, I do have a vehicle, so I'm not limited by that. So if you're, wondering how far is too far, send me an email or chat with me and we can figure that out. Any other questions on the field guide? Well, I can answer things via email too. So if you're thinking of something tomorrow, can't find me, send me an email. But I think we left off here in looking at, microbes are really quite good at exchanging DNA with their neighbors in their habitats. And we were talking about how plasmids can be transferred. Remember, so there's 3 ways we talked about it. What's the way that plasmids are famously transferred? You have the option of transformation, transduction, and conjugation. Which of those 3? Conjugation. It's actually up there. So they, they are studying this pretty intensely because from the human perspective, some of these things that are listed here can be quite worrisome, particularly antibiotic resistance. And so if you talk to a medical microbiologist, they're quite concerned about how easy it is for antibiotic resistant genes that are on plasmids, how easy that is to transfer amongst a population of microbes. And the idea here is, if you're in an environment where there's lots of antibiotic use, that's a selective pressure then on microbes. And so it's a outside force that's acting on their evolutionary trajectory. And so if they're surrounded by a bunch of antibiotics, it kind of makes sense that they'll hang on to those antibiotic resistant genes because it helps them survive. And they've done these studies. They've looked at it. If they get rid of, there was a case study where they were looking at penicillin resistance. And so they took penicillin out of rotation, so to speak, in that in that area, that geographic area, so people weren't getting penicillin, and they could track the decline in resistance against that. So the bacteria became more sensitive to it again. And so again, this idea of if you have an environment where there's a lot of antibiotics that can kill the bacteria, if they gain a gene that makes them resistant, they're going to hang on to it, pass it on to their progeny. And so this is something that they're looking at. Lots of different scientists are thinking about this. You know, they're starting to realize that antibiotic resistant genes are in our environment. And here they're looking at, you know, what's going on with antibiotic resistant genes in natural environments. They're looking at mineral surfaces. Remember, we talked about mineral surfaces, how that can be a good way for catalysis and form, you know, during the evolutionary process. So they're looking at soil, sediment, food, and lakes, and they're trying to see what kind of environmental habitats really allow for persistence of antibiotic resistance genes. So this is something that even airborne. So they're looking at cattle feed yards. So if you are not aware of it, when probably the biggest source of antibiotic use currently is in the agricultural realm. Growers, or I guess you don't really grow animals, what do you call them? I mean, people who raise cattle and pigs and chickens, and especially in the big operations, it's very, very common for them to give antibiotics as a routine part of that animal's life, even though they're not sick. Because they found out if you give antibiotics to animals, it actually allows them to put meat on their bones faster. And so they grow faster. And it is, you know, from the farmer's perspective, they are probably thinking it's helping prevent spread of disease, but not so much. And so cattle feed yards where all the cattle are together in small areas. You have airborne particulates from dust and other particles coming up, so they're tracking that too. Wastewater treatment plants, people flushing their antibiotics down the toilet or sink is another issue to consider. So this antibiotic resistance is not going away. It's been a concern for quite a while, and so far it's still a concern. They're tracking it. In this study, they were looking at over 20 weeks of persistence, but I think they're going to find longer stretches of time the more they look at this. So this idea of antibiotic resistance is a concern. On the other hand, there's some good stuff going on here too, in the sense that a lot of the plasma genes for degradation of toxins in our environment that are toxic to us and to other organisms. So that can be a good thing to have microbes breaking those down. And we'll talk more about that. So this ease of transferring genetic material has been documented quite well. And again, you have to look at particular habitat those microbes are living in. If there's antibiotics in that habitat, then they're going to keep those resistance genes. If that selective pressure is removed, the antibiotics somehow are going away from that habitat, then it makes sense that they might purge that because it's extra resources they have to apply to it to keep it going and keep it maintained. So it's a selective advantage in certain habitats, and they actually tend to use the word infectious because of how easily it's spread throughout a population, like in a hospital environment. And they now know that this idea of sharing DNA can go between pretty distantly related microbes and even other organisms. So they've been looking at how, you know, bacteria and fungi are interacting with each other and sharing genetic information. Bacteria and plant cells, that's well documented. And so this is not full, but basically what I'm trying to show in this table is that in the bolded areas, they have what's going on with the interactions. So you have domain archaea to bacteria, genetic sharing going on, bacteria to bacteria, bacteria to archaea, and then here you have bacteria to eukaryote. So they're jumping domains. You know, domain bacteria, very unusual, and yet you're able to have this transfer of genes. And then, so this is something that is leading, at least in the microbiology world, is leading to think a little bit more about how to consider evolutionary processes and phylogenetic trees. So this is a simple mock-up of what they're proposing. We should be thinking more along these lines for phylogenetic trees instead of the one that I showed you previously that was a little bit more bare bones in terms of it going off and branching off. Here, they're trying to get across the point that you have a lot of horizontal crossing going on. And so this evolutionary process, they're saying, is being heavily influenced by horizontal gene transfer, and we need to consider that. And so here again, typically this top picture is how they might depict speciation going on in the evolutionary process. But if you talk to a microbiologist, they would point more to this on the bottom, where you have a lot of horizontal crossing here. And so if you talk to microbiologists, they even get a little speculative about how do you define a species, like a microbial species, because there's so much of this crossing going on in their lifetime that it can be very challenging when you start thinking about how do you define a species when they're doing so much of it. And so it's an interesting discussion, and you can discuss it with other microbiologists. It's a little hard to have this discussion with someone who's not a microbiologist because it's a hard concept, but it's getting out there. I see it a little bit more in the common literature, but I don't know if you've ever been introduced to this idea. Do you remember in like basic evolution class, do they talk about it? Oh, I see some nods. Okay, well, I hope so, because this is becoming more and more out there, and people are saying we need to think about this. This was one that was published a while ago, back in 2016, but I really like it because, again, it's showing, if you look at this in totality, I mean, look at the whole top portion, that's all domain bacteria. So this is a phylogenetic tree. This is, all of this is domain bacteria. And then look at the eukaryotes, just a little bit over here. And so it's a pretty nice visual showing that they kind of rule the planet in terms of sheer numbers and different species. And there's so much yet to discover. So it's an ongoing, never-ending process. This is a paper that was published in 2018, and again, looking at combining the idea of horizontal gene transfer again with mineral surfaces. And so they're really starting to look at that. They know that DNA can get stabilized on minerals, and that allows DNA to persist, which means it will be around longer for microbes to pick up in the transformation process. And so they're really saying, we need to think about this back when we have those ancient photocells, that could help explain part of the story of the evolutionary process on Earth. And this is a figure from that paper. And here they're showing kind of and this idea of DNA on the mineral surfaces. And at least with this picture, they're really focusing in on the ocean floor, and the idea of black smokers are the term given for certain types of hydrothermal vents in the deep ocean floor that spew out superheated fluid from essentially coming across underneath. And so you have very, very hot things coming through, and microbes actually live on the surface of those black smokers, and with that, all that hot, rich mineral water flowing past them. They mentioned stromatolites in more shallow areas. And so they're looking at different environments and looking at the ability of DNA to persist. They're saying that's helping further explain the evolutionary process on Earth. So lots of interesting work going on, and of course, it'd be great if we could take a time machine and go back and actually see what it was like, but unfortunately, we can't do that. One thing you need to be aware of, though, is that bacteria are a little different when you think about their DNA compared to, what we have. They're what we call haploid. So we're diploid. We have 2 copies of genes, one from your dad, one from your mom, all right? They don't have that. They're genetically haploid. They have 1 copy of each gene. So that is a little dicey, right? If that gene becomes mutated to the point where it can't function correctly, and if it's an important gene, that could be a problem. So they do have strategies that they can pull out and use. They can go through a process called gene duplication, where parts of their chromosome, certain genes, can get duplicated. And they find, at least in lab-based studies, that helps. If you experimentally mutate a gene, there are options that they can use with this gene duplication. I won't go into detail, but basically they can duplicate a gene, and sometimes it just stays, with its original function. Sometimes they can duplicate a gene, and then they'll start, for lack of a better way to describe it, they kind of play around with it through mutation events. Of course, they're not doing it purposely, it's just natural mutation going on. But if those mutations lead to something that's useful, then that gene can become kind of a new thing for the microbe. And so there's these kinds of processes going on too. It's not just horizontal gene transfer. They can kind of play around with some of their duplicated genes, and that can lead to some interesting phenotypes. So we have gene duplication that can also help with speciation to some extent. They're not quite sure how extensive it is yet. This is kind of there's a lot of mystery to that whole process still. We certainly know about mutations, that you can have mutations going on in the genome sequence. And then, that can happen just through when they're going through replication. There might be a mistake when they're trying to copy their DNA. It could be some outside force like ultraviolet light. That's commonly, by the way, what they use in research labs. They often hit microbes with ultraviolet light. Sometimes they'll bathe them in a certain chemical that they know can mutate. And so there's those types of ideas. And then horizontal gene transfer is a big one. Okay? More and more realize how much that's playing a role in the evolutionary process. And don't forget about gene loss. So with eukaryotic organisms, that's not talked about too much as part of the evolutionary process, but with microbes, it's a big part of the process, because we can have gene loss occurring to the point where some of these microbes become dependent on whoever they're hanging out with. And so there's some obligate parasites, for example, that if you try to separate them from their hosts, they won't make it. So they've gone through so much gene loss over time that they fully depend on their host to survive. So, yeah, exactly. And so, unfortunately, there's a lot of parasites that fit into that too. But this idea that when you have, and we'll talk more about this when we get into microbe interactions with other organisms, but you can have this coevolutionary process where they're each evolving with each other, and they're each influencing each other in that process. And so as they're doing that, it can lead sometimes to this state where the host is providing all the necessary day-to-day nutrition to that symbiont, and they're dependent. So just keep in mind, again, it's adaptive to their habitat, right? It has to be, you have to think about it in context of their habitat. And if you have a habitat that changes dramatically, that their genetic composition might actually be might harm them in the sense that they don't have what it takes to adapt to that new habitat. And so again, you always have to put it into context of what's the current habitat. Is that mutation or that horizontal gene transfer event going to be a good thing or not for that microbe? So can they survive and adapt better? But adaptive mutations certainly do improve the fitness of an organism, and that helps to increase its survival, helps it then pass that all those genes on to the next generation. So basically, we're thinking about, yes, you have the kind of tried and true idea that's presented in basic evolution classes, that you get periodic selection going on to mutation events for favorable trait. Certainly, we got that going on, but don't forget about horizontal gene transfer. And we're looking at different habitats, different organisms in them. So as we're moving forward in this class onto the next topics, I really encourage you to have this frame of mind that, quote, nothing makes sense except in the light of evolution. You have to think about the evolutionary process and look through what's happening, look through the lens of the evolutionary process as you're trying to figure out why is a microbe, are there microbes here, what are they doing, what's their genetic makeup, what's their phenotype. And so we'll try to keep that in mind as we progress. One other thing I wanted to mention in terms of the evolutionary process, I put, whoops. I put in EduCat, there's a small slide set on Lenski's grand experiment. So I'm not going to go over that right now. This is more typed up so you can read it on your own. But this is something I talk about it in Gen Micro too. It is, whenever I'm like out in the community or with friends or something and talking to non-science people, and they talk about evolution, and they're not really either aware of the process, or maybe they don't think it's a thing. It's very easy for me to start talking about microbes and that you can actually see evolution in action in your own lifetime. You don't have to wait. With humans, evolutionary process is very slow, but in microbes, you can do it within a matter of months, and you can see evolution occurring and working with them in the lab. So this is an experiment that's kind of along that line, where this was done down at Michigan State University, and he was able to show a pretty significant effect in terms of, he worked with E. coli and how it evolved to become a special kind of subspecies based on how they were interacting with it and manipulating its environment. So please read through that. We'll talk about it a little bit after you've had a chance to read through it. Okay. Let's get into the next topic. It's posted in EduCat on ecological physiology. Okay, microbes. They're tiny compared to us. So we have to think about that limitation and then think about some of the special ways that they try to eke out a living. This is just a visual trying to show you some of the more common body shapes of microbes. Coccus is spherical, rod shaped. Sometimes in the older literature, you'll see the word bacillus instead of rod. They might use the word bacillus with a small b, and that means rod shaped. And so if you're reading some of the literature, don't get confused by that. That's the older term. They've switched to rod because this term bacillus is confusing because there's a genus of bacteria called bacillus with a cap. And so that's kind of confusing. And so they decided to switch and start calling it rod shaped instead of bacillus. Okay. Spirillum shape is another one, spirochetes. And then have some interesting where they have some extra kind of appendages, so to speak, like stalks that they use. Often, if you see something like that, you've taken a sample from a watery environment. They tend to come back to out water, flowing water. They tend to have stalks that latch onto the surface and then kind of dangle in the current and wait for things to come by. So those are some of the basic shapes. And there's many different species that are coccus shaped. And there's many different species that are rod shaped. So it's not like you can slap a sample onto a microscope slide, look at it and go, oh yeah, that's so-and-so. You can't do that. It's not that easy, unfortunately. So when you look at bacteria in general, they're small, they're tiny, and you're gonna need to get used to that in lab. If you've not had a microbiology class, when you work with your microscopes, you're going to be using oil immersion and you're going to be hunting for things that are even with oil immersion lens that magnifies pretty high level, you're still going to be looking for tiny things. So it takes a little getting used to. So you can see two different spheres, a small one and a big one, and you can see that your surface to volume ratio is pretty decent if you're small. If you're big, your surface to volume ratio goes down. So one reason they think microbes maintain a small shape and have not really gone beyond that over the evolutionary process is that they need a large surface area. They need that. It's important to them. And I'll help explain why that's important to them. So keep in mind, the smaller you are, you get some nice surface area. Keep that in mind. All right, well, here, I guess we're getting into it. It's the prokaryotic cell thing. When you look up the definition of prokaryotic, they'll say no nucleus, no internal organelles. And that's mostly true, no nucleus, but some microbes could have internal organelles. You could define it that way, but we are reluctant to do so. Okay? All right, so if you don't have internal organelles, then where do you put all the stuff that are needed, like our mitochondria? Super important to us. That's an internal organelle. We have to have it up and running because that's how we get our ATP. So what do microbes do that they don't have mitochondria? I mean, there's free living ancient microbes that want mitochondria, but where do they put all their stuff in order to generate their ATP? They don't have all these nice little compartments inside them where they can have that. And so what happens is the cell membrane gets this huge task of not only keeping a selective barrier around the microbe, but now you got to stuff all the proteins and enzymes in there too that are necessary for ATP production and many other processes. So the cell membrane is hugely important to the microbe. It's a workhorse for sure. It's doing many different things. And so that is one reason why they point to the idea that microbes really haven't evolved to get bigger. They need that large surface area because they don't have internal organelles. Now, having said that, depending on their habitat, one thing to appreciate is that bacteria can vary their size within their own lifetime. So if they're hanging out in habitat And everything's great, flush nutrients, lots of water, they're living the high life. They'll be their so-called normal size. If they start going into tough times, they can actually shrink their size, which again increases their surface area, which then can allow for certain things like better transport of nutrients back and forth, waste going out. And so you have this idea within their own lifetime, you can see that you'll have very shelf size. Keep that in mind. And it's tied quite tightly to feast or famine conditions. Here you're looking at largest on top down to smallest on the bottom. So, right here you see Thiomargarita namimbiensis. Very mouthful. Thiomargarita. I got all excited when I saw that genus name. I'm like, oh, I love margaritas. I wonder, did they after, because you can name, if you discover a microbe, you get the option of naming it. So I'm like, oh, I like this scientist. She likes margaritas, whoever that. But Thiomargarita. That's margarita. Margarita, I think, means pearl. I'm not remembering. Thio is sulfur. So sulfur pearl of Namibia. And that's where they discovered it. Namibia. And so it's spherical. So that's why they thought pearl. But anyway, so I'll put it here. So that one is quite large. That is what we would consider an anomaly. 750 micrometers, that's huge. And then here's another big one, Ecological. You can think what kind of organism it might be associated with, this type of name. Fish. Yeah. They found it in the guts of a sturgeon fish. So again, they made it, to honor that. But that one's also quite large. And then you go down, you're getting smaller and smaller and smaller. And if you go all the way down to mycoplasma, mycoplasma pneumoniae, which causes there's several microbes that can cause pneumonia. This is one of them. Mycoplasmas in general are very, very small. This one is if you've ever had walking pneumonia, if your doctor told you you have walking pneumonia, that's what you have. And I had that once when I was in high school, and the doctor said, oh, well, he didn't know it was that. He said, you have pneumonia. So he gave me penicillin. And those penicillin pills were like horse pills. It was, ugh. So I took that whole treatment, didn't get any better. And then went back to the doctor, like, oh, you probably have mycoplasma pneumonia. Mycoplasma is not sensitive to antibiotics, because at least to penicillin, I should say, because penicillin attacks the cell wall of bacteria. And this one, mycoplasmas, don't have cell walls. They're weird bacteria. They look more amoeboid. They have no cell walls, so they can change their shape, and they're tiny. Very interesting group of microbes, but you can see 0.2 micrometer. And pathogenic. So yeah. So you see there's a whole range. Let me just show you a picture of Thiomargarita. So this actually does have what they refer to as vacuole, which is a compartment. So, and we talk about vacuoles in plants as an organelle. So again, this is what I mean. We're not quite there yet to declare that microbes, at least some of them, can have internal organelles, but I think someday we'll get to that. So here is what it looks like on the microscope. So it does look like a turtle necklace in some regard. And so here is out a little bit, but quite large. And then Epiphysium, I'm just going to go through this fast. This is all in the PowerPoint and EduCat. Yeah, the surgeonfish. So this one has, again, many infoldings. So when they looked at this one, remember, as you get big, your surface area drops. And but they solved that problem because they said, yeah, we're big, but we're still going to have good surface area because we're going to have infoldings of a membrane. And whenever you do that, you increase the surface area. And so even though they're big, they solve the problem of making sure that they do still have good surface area. So here is the size difference. E. coli is black dot compared to just part of E. officium here on the slide. Here's the eukaryotic paramecium. So this one breaks the mold. If you break open a textbook, freshman level textbook, they'll tell you that prokaryotes are smaller than eukaryotes. And clearly this one exception. As with, and pretty much anytime in science, there's always exceptions. You can't be so, black and white about it. So here you have on a pinhead, see all of these are efficient. So again, just trying to reference. So if you have really good eyesight, you can see this with the naked eye. Okay. So a couple of things we need to think about as we move forward in this class is a little bit about their genetic makeup. So I already told you they're haploid. And so when you are in the microbial world, they often talk about open reading frames. You'll hear that a lot in the literature. Read that. And basically, you can equate that to a gene. When you look at different microbes, they have discovered that you do get quite a range in genome size. So their chromosome size can vary depending on who you're thinking about. And the other really confusing thing is that you can see strains of microbes that are related to each other. They can have some wildly different genotypes and phenotypes. So I would not want to be an evolutionary microbiologist. I think it would drive me batty when you get into some of the fine scaling of what is a species, because it can be confusing. So let's take a peek at some of these ideas in this table. All right, the genome size, they don't have it nice organized, do they? No. Very not like compliance with so you have megabase here, genome size, and then they have the open reading frame. That's like the number of genes. So Agrobacterium tumefaciens, that second one down there, you can see it's got a pretty decent genome size, and it's a pathogen. So phenotype and habitat, it's a plant pathogen, very famous plant pathogen. This microbe likes to hang out in the soil. We'll talk more about that one as we get into plant microbe interactions. So let's look at a couple others. Anabina, the one right below it, is photosynthetic. You can see its genome size is quite large. Generally, that's a good rule of thumb. If you're photosynthetic, you tend to have higher genome size and open reading frames. That's a lot of stuff to build when you look at the photosynthetic machinery. You can also see that it can hang out in soil and water. The one below it, B. cereus, get it? So Bacillus cereus, and then whoever named it had to have a sense of humor, because usually, you don't say the full genus word after, you introduce the, Bacillus, and then you just abbreviate, and so then you say B. cereus. So microbiologists can have humor, and again, you can see it's in the soil, and again, genome size is pretty good. Look at this one, Bradyrhizobia japonicum. This one again is in the soil. It's nitrogen fixing, which is a very, very special trait. And if you are a nitrogen fixer, again, you have to have a lot of special genes. involved in order to do that. You can see that it's quite high in terms of its open reading frame and genome size. So you start to see a pattern here. Things that are in water and soil tend to have pretty decent genome sizes. So keep that in mind. Soil habitats, water habitats can be quite complex. And so we have to keep that in mind. And on the other hand, look at Carcinella. Right there, Carcinella rudii, 0.16, only 182 open reading frames, and it's an obligate endosymbiont of a psyllid, which is an insect that feeds on plants. So here you have a plant pest that has an endosymbiont inside of it, and it's obligate, which means if you try to get rid of Carcinella and grow it on its own, it won't be able to survive. And probably the insect would die too. Usually that they symbiotic process is so tight that they need each other. Okay, so you can see there's quite a range of genome size and open reading frames, and it's highly driven by their habitat. So their habitat is a selective force that's directly, all the way down to the genome level. It's influencing their genomes, genome size, open reading frames. This is more of that table. Here's a Mycoplasma genitalium, intracellular human parasite of the urogenital tract. Again, you can see a very small genome size. But again, if you're in a more complicated habitat like soil or water, you tend to go up. Now here's an exception way at the bottom, Thermotoga maritima. You can see has a pretty small 1.86, but it's a hypothermophilic organism. So when you get into some pretty hostile environments, hypothermophile, hyper, that means way above, right? If you're hyper, you're way above. You think of what thermal, what they might be referring to there. Temperature, like a thermos bottle, In this case, hyper, so they mean very hot. Philic, you see that on the end of the word? That means to love. So what they're trying to imply there is that Thermotoga is actually well adapted to very hot conditions. It thrives in those conditions. It doesn't just survive and eat out a living. It's that's what it's adapted to. So use the word philic, that's what you're getting across. Okay, so you can see that you have these, clear differences in genome size and quite a range depending on the microbe. And so usually the rule of thumb is the more complicated the habitat, the bigger the genome size, so that they have lots of options to play with in terms of what genes to turn on, what genes to turn off. Here you're looking at the idea of what is all that genetic information for. And so the big picture idea here, the bolded words is what I want you to get across here and think about. So they're kind of grouping them in terms of what genes are necessary for information storage, processing. And so this is just looking at Pseudomonas putida. Pseudomonas putida is a well-studied lab rat, so to speak, in the microbial world. And so cellular processing, they have it grouped for that as well. Metabolism, that makes sense. Genes coded to that. And then you can see there's some that are we don't know. We don't know what these genes do yet. And so, there's a lot going on in the microbe. It's kind of amazing when you think of how small they are, but how much they have to do to stay alive. So I like this mock-up where they're showing a microbe and they're trying to show the complexity of what's going on inside that microbe. I believe this is also taken probably from Pseudomonas, because I see they have biodegradation, xenobiotics. Pseudomonas species are really well known for cleaning up toxins. So I think this is a nice visual. It just shows you how busy everything is going on. There's a lot going on in the cell. Here's another one. I like this image because it's really hammering home the point of how important the cell membrane is. Look at all the stuff stuffed in the cell membrane. And you don't see a bunch of organelles in there because they don't have them. Okay, so they have to put all these different proteins and enzymes and transport proteins. You have to stuff them in the cell membrane. So again, they need an intact, healthy cell membrane. That's super important to them. And so for a lot of antibiotics try to target that, by the way, because that can be very disruptive. Okay, so we'll look a little bit more at this next week. And then the next topic we're going to get into is a little bit of physiology to help you understand how they can adapt to these different habitats.